Okay, so this is part 2 of chapter 4. We did this part already, but just because the next part is related to it, so I'll go through it quick again. We talked about this before, and we said that the cell envelope in general consists of 2 parts: cell membrane and cell wall. Remember that the cell membrane is in every cell on Earth, right? Any cell should contain a membrane, right? But not all cells contain a wall. We agree on that? Every single cell, human, animal, plants, bacteria, eukaryotes, prokaryotes, anything, they all have to have a membrane. Otherwise, you don't call it a cell in the first place, right? Does it have a wall or a cell wall or not? It depends, and we will discuss that. So, regarding the bacteria, when we talk about the bacteria, for this part, if you add a stain, and we will go through the details of the gram stain, and this is medically important for treatment specifically. The types of bacteria can be gram positive and gram negative. Gram positive, they have a cell membrane that's thick and primarily peptidoglycan. The negative ones also have the same thing, but it is thin and have an outer membrane. Did you get the difference? Both of them have peptidoglycan, right? But the gram positive have it thick. The gram negative have the same thing, but thin and additional membrane. And that will make the difference. So peptidoglycan, it's peptido and glycan, so it's a glycan that's attached by peptide. But the teichoic and lipoteichoic acid is something in the cell membrane of the gram positive ones. Okay, so we need to remember this part. So here is a gram positive, and you see, look how thick this is, the brown. Okay, so if you look at this, the whole thing is called, what is the whole thing here? Cell, the envelope, right? This part of it is called membrane, and this part is called the wall. So bacteria have a wall, but all cells have a membrane. Every cell have a membrane. But look at this. This is characterized by, this is thick, which is peptidoglycan, which is the wall, contain peptidoglycan, and teichoic and lipoteichoic acid. Then if you look at the negative ones, they also have peptidoglycan, but they have an outer membrane. They have additional membrane that contain lipopolysaccharides. And I mentioned that this outer membrane makes a huge difference, and it will make the gram-negative cells kind of more aggressive. Why? Because this lipopolysaccharide, the lipid part of it, acts as an endotoxin. And this can lead to toxicity in the patients, and can lead to something called toxemia, like toxins in the blood. And it also can block the immune response. Doesn't that make it more virulent and more pathogenic? It also contains something called purine, and purine is coming from pores. So purine is a protein that regulate what goes in and what goes out. So 1, 2, 3 factors in that membrane that make the gram negative more aggressive. Look at the gram negative. Look at the peptidoglycan. You see that brown? This is thin, and outside of it you have an additional membrane that have what we talked about, which is lipopolysaccharide and purine. So this is a comparison between both of those. This is the membrane, this is the wall. This is the membrane, this is the wall. This is positive and this is negative, right? The positive have this thick peptidoglycan, the negative have thin peptidoglycan, and this is the additional membrane out here. This is an additional membrane that contains the lipopolysaccharide. So here is the comparison again. The gram positive have one layer that's thick and contain teichoic, lipoteichoic, which is not the case for the negative. The negative also have peptidoglycan, but it does not have teichoic and lipoteichoic. It have an additional membrane, and this membrane contains lipopolysaccharide with porine protein. So this is obviously thick, thin. Even though the gram-positive have a thick layer, but it's easy to penetrate. Even though gram-negative have that thin wall, but it's less penetrable. Why? Because of different reasons. Most importantly, that additional membrane, which contain the porine. So which one is not found in the gram-negative? Teichoic acid, and the other one is called lipoteichoic, right? We don't have it in this question, but just to remember it. And can you tell me which one of those is in a gram-negative, not in a positive? It's two answers. Yes. Porins and lipopolysaccharides. So you're following. Okay. This is the answer. So this will bring us to the gram stain. The gram stain is a stain that we added to the bacteria, and some bacteria will take it and retain it. Others, and this is called the gram-positive. So gram stain is a violet colored stain. When it stains something, it becomes purple, okay? The difference between the gram-positive and gram-negative is not that's taking the gram stain. This is not the difference. It's keeping it. So the gram-positive will take it and keep it. The gram-negative will take it and lose it, okay? Is that clear so far? And this will help in the diagnosis. Like if I take a sample from your sputum or something, I can look at the bacteria, I can add the gram stain and do different steps. So if it take the gram stain and keep it, I will tell you this is gram positive. And what color is it? Violet purple, okay? If it does not, if it take it but lose it, and then when I add another stain, it will become red, that will be the negative. So these are the steps. How to do that, how to prepare, and just general idea. It's not like all the details. Here's what we're going to do. I have a bacteria, okay? And I wanted to know if that gram positive or gram negative. Steps of staining. It's not just a gram stain. The first thing is we add the gram stain, which is, or the crystal violet stain, which is the primary dye. This is the first thing that we're going to do. And if you notice here, both of them will take it, right? Gram positive and gram negative will take it, both of them. But if you noticed further, you will see that this is thick, so it takes more, and this takes less. But anyway, both of them will take it, and this is not the issue. So the first thing is crystal violet, both of them take it. Step number 2, you're going to add something that's called iodine, grams iodine, and this is a stabilizer. So it's supposed to stabilize this color so it doesn't go away. Both of them will take it, but when you go to step 3, you will add alcohol, and alcohol that is decolorizer. So it should remove that violet or purple color. If you notice here, look at this, the gram will retain the violet color. It's not going to lose it, even though the alcohol is a decolorizer. So it's supposed to remove the color. In the positive, it didn't remove it. But in the negative, you see the color is removed. So it didn't keep it. Why it didn't keep it? What made the positive keep it? There is something in the gram positive that keep it, that make it keep it and doesn't lose it, even if you add alcohol. Teichoic and lipoteichoic. Okay? So teichoic and lipoteichoic, because it's only in the positive, they will not only take it, they will retain it. Even if you add alcohol, yes, even if you add alcohol. This is something that's the negative is lacking, so it will not keep it. Even when you add the stabilizer, it still lose it. Now you will add additional dye, which is called, it's a counter stain, the opposite. And this is called the sifronine, and sifronine is a red dye that will stain only the bacteria that lost the color. Which one lost the color when we added alcohol? the negative, so the negative will take it. The positive didn't lose it, so it will not take it. At the end, you see how this is what you're actually at the end of preparation of gram stain. This is what you're actually going to see at the end. So when you see something purple, that's positive. When you see something red, that's negative. But you just know how the steps work, right? How did you get to that point? At the end, the most important to remember is positive is purple, and the negative is red. So this is summarizing all what we talked about. Crystal violet added, both of them take it. You add the iodine to stabilize the color. The color is supposed to be stabilized, but when we add alcohol, you see the difference. The positive keep it. You cannot decolorize the positive, but you can decolorize the negative. When you decolorize the negative, it lost the color. So when you add the safranine, which is the red dye, it's going to take it. Okay? So this is generally speaking when we talk about the cell wall. Some exceptions to these rules are, we have two exceptions. Number one, there are some bacteria that does not look exactly like we talked about. They have something else, something additional. Like some gram-positive cells, they have a wall that contains lipid called mycolic acid, and you need to remember this. Lipid called mycolic acid. They call it the cord factor, and I will tell you why they call it the cord factor. And remember all of this. Some gram-positive bacteria have mycolic acid, and you call that mycobacterium. Where the name came from? It's a bacteria that have mycolic, so you call it mycobacterium, mycolic acid. Did you get where the name came from? Mycobacterium. And this mycobacterium, because of the presence of this mycolic acid, they have a high degree of pathogenicity and resistance, and they call it acid-fast bacilli. So when you add the acid-fast, it will take it. When you add the acid-fast stain, it will take it because of the presence of the mycolic acid. So what do we need to know about these cells? Again, the non-typical cells. These non-typical cells, examples are Mycobacterium and Nocardia. Remember this. These atypical bacteria, Mycobacterium and Nocardia. You have two to remember. What are these two? These are gram-positive, but they have additional thing. Just like any gram-positive, they have a thick peptidoglycan, they have teichoic, lipoteichoic, that's all okay. But they have additional thing, which is called the mycolic acid. And because of that mycolic acid, we call it mycobacterium. What's the difference, or what does this do? It does the following. Number one, it makes the bacteria looks like a cord shape. And this is why you call it the cord factor. Okay, it looks like a cord like this. Okay? So why does it look like a cord like this? Like compressed and long. Why do you call it cord? Cord factor, because it makes these bacteria looks like cord shaped, like this, elongated and like this, looks like a cord shape. So this is mycolic acid. It make it like a cord. This is one. Number two, it make it very pathogenic and very resistant. Very pathogenic, very resistant. And it's stained with acid fast. So when you hear acid fast bacilli, what we're talking about here? You're talking about mycobacterium, basically, most importantly. Why? Because it's acid fast. Why is it acid fast? Mycolic acid. Okay, did you get this point? The other type that's typical, even though it is a bacteria. Bacteria have a cell membrane and a cell wall, right? We agreed in the beginning that the bacteria have cell envelope, right? And this cell envelope is membrane and wall, right? Agree on that? So this is, any bacteria should be like this, except for this one. Mycoplasma does not have a cell wall. Still count as bacteria? Yes, it does. Why? Because the rules apply here. It's prokaryote, it does not have a nuclear membrane, it does not have membranous organelles, and so on. So everything apply, so you have to categorize under bacteria, but it's not typical. We call it the atypical bacteria. So the typical bacteria contain what? If you got what I mean here. The No, typical bacteria in general, mycolic acid in these type of bacteria, which is the mycobacterium. But any bacteria in general should have a membrane and a cell wall. If it has a membrane and cell wall, I call it typical. All of them have that. If it does not have a cell wall, you call it atypical. So mycoplasma is atypical for reasons. Okay, do you get this?
Yeah.
Mycobacterium nocardia is atypical. Why? Mycolic acid. Mycoplasma is atypical. Why? No cell wall at all. And the reason why we call it mycoplasma. It's like a plasma without a wall outside. If that helps you to remember it, but you have to remember these three names anyway. And you need to know why Mycobacterium nocardia are atypical and why Mycoplasma is atypical. Okay? Now, if you look at the inside of the bacterial cells, do you guys remember in A and P, do you remember these organelles inside? The chromosomes and the Golgi apparatus and ribosomes and all that stuff? Almost, or very close here. So cytoplasm is the first thing, just like our cells. It's mainly water, and you have some organic molecules inside that water. It's gelatinous a little bit, but contains just like our cells. You have some sugars, amino acids, and salts, just like our cytoplasm. So this part is just like our cells, cytoplasm. Inside the cytoplasm, you have nucleoid. Nucleoid is not nucleus, it's nucleoid. Oid means similar to, right? Oid means similar to. It's similar to nucleus, it's not. It's just a genetic material. Does it have a cell membrane? Does it have it does have a cell membrane. Does it have a nuclear membrane? Is the bacteria prokaryote or eukaryote? Prokaryote. Prokaryote have a nuclear membrane or not? You guys forgot. Okay. Does it have or not? Nuclear membrane. It does not. Okay, great. So it does not have a nuclear membrane, but it does have chromosomes. If it does not have chromosomes, it does not count as a cell in the first place. How does that chromosome looks like? A single circular and double-stranded molecule. Okay? Just one that is double-stranded and circular. It looks like this. Okay? This looks like this. One circle, small, and double-stranded. So this is the chromosome itself. Okay? And this is have the genetic material that's needed. So this is the chromosome part. Chromosome is single, it is circular, it's double-stranded, so they have just one. Plasmid, on the other hand, is something additional. Looks like it, but a lot smaller, and it is free. And this is used for genetic engineering. They take it from one and they can add it to the one. They work around with that plasmid, which is easy because it's very small, very tiny, it's free. So they can take it from there and use it for genetic engineering. What else do we have? We have the ribosomes. Do you remember the ribosomes in the cells? What do they do? Proteins. They just make proteins. The other structure that's in the bacteria, which is something that's different from our cells, and this is called the inclusions and granules. Inclusions and granules, simply storage. Just storage. Storage of what? They store additional nutrients just in case. If there is no nutrients around them, if the environment does not have the nutrients that they need, they can use it. They have it in different shapes and different contents, but this is their storage. Something happened, they don't find the nutrients, they start to break down these granules and inclusions, and they start to use what they have. So what's the function of inclusions and granules? One word: storage. Storage of nutrients. The other thing is something called endospores. And endospores, we talked about it before, that this is if the environment is really bad. Remember when we talked about like TB? If somebody spitting a sputum on the floor or something. It's dirty and everything, and it can stay there for years, right? And the only way, it's very complicated to get rid of it. You clean it, it doesn't go away. You use the disinfectant, doesn't go away. There are ways to get rid of it, but it's extremely hard to get rid of it. So the hospitals that are specialized for TB, they know how to get rid of it. It's a pressurized treat. This is the only way to get rid of it. But other than that, it will stay in there, no matter how much you clean, because endospores are very resistant. Okay, they are very strong. You need to remember that the cells that make the endospores are gram-positive ones. Okay, gram-positive bacteria. Some of the gram-positive bacteria can do endospores, and the endospores are resting, not metabolically active. Okay, they don't do anything. Like, you know how the hibernation works? Like, you stay like this, you don't do any activities. Just wait until the condition gets better. For them, the condition gets better means they go to somebody else. They enter the body of another person. This is improving the condition. As long as, like, they are in a floor where it's dry, there are no nutrients, and the weather is bad and everything, so to them this is bad. They will stay at endospores. So endospores are very resistant to the environment. And is it metabolically active or not? Not metabolically active. It's resting. Okay. So this is the endospore. Generally speaking, the life cycle of those types of bacteria, not all bacteria, just this type that can make the spores. It can be in a vegetative form, and vegetative form is the metabolically active. These are the ones that can grow. Obviously, this form, if they have the suitable environment, which is most of the cases, or they can switch to endospores if the environmental conditions are not good, if it is bad. And they can survive for a long time. Believe it or not, they can survive for millions of years. And that's why it's never been eradicated. So when the vegetative cells change and become spores, you call that sporulation. And even the name give it away, right? Sporulation, spore, to change to spore. Germination means they switch to become the vegetative growth. So just switch back to the vegetative, which is just the metabolically active ones. Okay? So what sporulation means? Change into spores. And what germination? Change to the germinative form, which is just a regular form. So if you look at this, no big deal out of it, but just it's showing you exactly what happened. So this is just a normal cell, vegetative, and now the environment is bad. What do you mean by bad? Dry, you don't have nutrition, the weather is bad, the temperature is bad, whatever it is. We call it adverse environmental conditions, bad conditions. So what they are going to do, the chromosomes is going to duplicate itself, then it's going to separate into two pieces. The spore means front. The front part of the spore and the rest of it will stay, and we call it sporangium. Then the sporangium is going to surround the spore with additional layers. So now you have the early spore and the rest of the sporangium. Then the sporangium is going to add more layers to the spore part, and then it will disintegrate, means you get rid of it. Sporangium. Sporangium surrounds the spore with extra layers and then disintegrate. So you get rid of it. You end up having this spore. So look at the structure of the spore here. You will see the inside the core that have the chromosomes, and then you have surrounding it, you have the cortex, a coat, and exosporium. This is how strong it is. It has 3 layers around the chromosomes, which is extremely hard to get rid of it. If something happened, like this is somebody swiping the floor or something, and he didn't know there is endospores here, it goes to somebody breathing, it goes into your lung, so this is going to vegetate, okay, and it will change to vegetative form again. Did you get this idea? Okay. So endospores, again, these are metabolically inactive. They are dehydrated because you got rid of the most of the structure. You just left with a little bit part of it. It has a thick coat, which is one of the layers surrounding it. It can stay. The longevity is really, really long. It can stay to 150 million years. It can stay for millions of years. If you try to clean it with anything that we know, it doesn't work. The only thing is pressurized stream that's 120 degrees centigrade. And remember this. Pressurized, not only under pressure, it's above the boiling. Okay? Which is 100. It's 120 and pressurized. And you have to do it for 20-30 minutes. So if somebody come do it for 5-10 minutes, it doesn't work. It's exactly like this. Pressure 120, half an hour approximately. Can you answer this question? There is one word that will give you the answer. Granules. Your clue here is store. Okay? When you see store, what store? Granules. Okay, now the bacterial shapes. We're getting into the bacteria itself. Okay, the bacteria will have different shapes, sizes, and everything, but we usually classify them according to the shape into the following. If it is spherical structure, we call it coccus. If it is rod-shaped, we call it bacillus. If it is helical, twisted like this, rod, it's rod but helical and twisted, we call it spiralium. Spirochetes is one of those that looks exactly like a spring shape. Okay? So spherical, you call it coccus. Rod, bacillus. Twisted rod, spiralium. So spirochetes is one of the spiralium. Is that clear? Spirochetes is one of the spiralium. A bacillus. What kind of bacillus? It can be coccobacillus or it can be vibrio. This is very important slide, by the way. Very important. Okay? Coccus is spherical shaped. Bacillus is rod shaped. Spiralium is rod but twisted. Is that okay so far? Okay. Now coccus spherical shape, we will go to the details. If it is bacillus, means rod shaped. Like what kind of rod shaped? Rod shaped that looks like this. Rod. But it has something like this at the end. Can you see this? You call that coccobacillus. It is a bacillus, but have something here that's more dilated that looks like coccus. Did you get the idea? Which coccus is spherical, isn't it? So this is coccobacillus. And this is short and have this plumb at the end. If it is curved like this, you call it vibrio. The most common type of vibrio is vibrio cholera, if you heard about the cholera before. Spiralium is something that's twisted. Twisted means like this. Is it twisted like a spring? This will be the spirochetes. So here are the different shapes. This is coccus. Obviously, this is round, spherical shaped. This is rods. This is vibrio. See how it's curved? And this is spiralium. Looks like a spiral shaped, twisted. Spirochetes is one shape that looks like a spring. Looks like a spring. Pleomorphism. Pleo means different. Morphism means shape. So pleomorphism is different shapes. This is one of the criteria of the bacteria, that one type of bacteria will have different shapes and sizes in a single species, one species, but it's not exactly like, not like a copy paste, not like exactly all of them have to be the same. Spherical like this, but it can be spherical that's a little bit bigger, a little bit smaller. That's okay within the same species, as long as spherical structure, we call it coccus in general, right? Spherical, you call it coccus. Big or small, it doesn't matter, because you will have different sizes. Arrangement. So we agreed on the basics first, right? It can be spherical structure, it can look like a rod, and it can look like a spiral shaped rod. This is just generally speaking. If it looks like spherical shaped, are you talking about one by itself? You call it just coccus. If it is two spherical together, we call that diplo, double. Diplo means double, right? So it's two. It can be four, we call it tetra. We know that tetra means four, right? Tetrad. Or it can be irregular clusters like grapes. Look exactly like the grape. We call it staphylococcus. And if it is in a chain, we call it streptococcus. Okay? So it can be 1, 2, 4 cluster chain. Okay? 1, 2, 4 cluster chain. 1 coccus. 2 diplo. 4 tetrad. Cluster staph. This is very important to remember. If it is a chain, and all of this is cocci, all of these are spherical structures, and the difference, just why it's happening like this is what happened. Here is 1, it becomes 2. Or it doesn't become 2. Like here is 1, become 2. Do they stay together or not? If they stay together, that will become a diplococcus. If it does not, it will become a coccus. So now it's 2. The 2 became 4. Do they stay together or not? If they don't, it will stay diplo. If they come together, that will be the tetrad, and so on. Okay? So the way they grow and duplicate and whether they stay together or not will give them all these different shapes. On the other hand, the bacilli can be just one bacillus. It can be diplo bacilli, like this. What did you call this? Diplococcus. How about this? Diplobacillus. 2 bacilli, 2. That's it. This is 2 cocci together. This is 2 bacilli together, right? We call it a dip. And it can also be a chain. Chain is like this. Coccus, 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 right? What did we call this? If it's a chain of cocci. Strato? Yes. So if it can be 1, it can be 2, it can be 4, it can be cluster, it can be a chain. So this is streptococcus, and this one here is the chain bacilli. On the other hand, it can be palisade. Palisade is something like this. This is rod shaped, right? So this is bacillus. Here is another bacillus on top of it. It looks different than this, right? Something like this. Okay? We call that the palisades. One bacillus, 2, that's diplo, this is a chain, and this is a palisade. Generally speaking, to classify the prokaryotes, you have to gather as much as possible of information in order to categorize and classify the prokaryotes. And we went through this before, but it's important to remember how they classify. They use all of these criteria. They look at it at macroscopic level. How does it look like? And we're talking about colony now, right? Like you hold it with your hand and you look at it. It looks this color and this shape, regular color, round color, whatever the color is, and we're talking about a colony now. So a huge amount of prokaryotes. The next step is look at it under the microscope and look at how does it look like. Is it spherical shape, rod shape, and so on. This is the microscopic morphology. and then you study the physiology. Do they ferment? They do not ferment? How do they breathe? How do they use their nutrients? How do they work? And so on. This is all physiology of the bacteria, meaning you look at how they function. Serological analysis, you look at the serology, and serology means studying the serum. Look at the serum and try to find what's happening there. What kind of antibodies come against them? Is it effective or not? And how they interact with our immune system, okay? Genetic and molecular analysis, you take the genes and you study these genes. So you put all these criteria together. Actually, they put it in a computer program. Get all of this. This is the macroscopic shape. This is the microscopic. Looks like this. This is the shape. This is how they function. And the computer program will tell you, okay, this is the classification. There is a manual that's called Purge's Manual. And this is used for that classification. It's called Purge. And they call it the 5-volume resource. All prokaryotes, they put it in different volumes, but basically the basic classification, according to the genetic information, is either archaea or bacteria. These are the prokaryotes. Archaea and bacteria. So volume 1 is archaea, volume 1 domain. And archaea is it is a prokaryote. It's a typical prokaryote, but the only thing different is it does not have a cell wall. Does the bacteria have a cell wall? Yes, it does have a cell wall, except one. There is one that does not have a cell wall. You still call it bacteria, which is called mycoplasma, right? But generally speaking, this is an exception. Generally speaking, bacteria have a cell wall? Yes, for sure. They have to have a cell wall. Beside the cell membrane that's in every cell, whatever it is. So it's a prokaryote that does not have a cell wall. So here is a prokaryote. It can be archaea, it can be bacteria, right? Does it have a cell wall? Yes, it's bacteria. Is it a prokaryote that does not have a cell wall? It is an archaea, except mycoplasma. It doesn't count toward the archaea. They always say that the archaea is, they give it that name of extremophils. Extremophils. Extremo means extreme conditions. Phils means they like or they are okay with this extreme circumstances. Extreme like what? Like extreme temperature. So they grow like close to the volcanoes. If it is really hot water, ice, water, it doesn't matter. They grow under extreme temperatures. Not only the temperature, they grow under extremes of pH, right? Like not around the neutral, not necessarily. Even if it's very acidic or very basic, that's fine. Even acidic, so they call it acidophils. So even with acidity, the prokaryotes, they die. Not this one. They are very resistant to acid. Pressure, when you pressure them, still resistant. If there is no oxygen, you call it anaerobe. An means no. Aerobe means air, and we're referring to oxygen. So even with the lack of oxygen, they can still survive. They can be one or two or all, depending on the type of archaea. And archaea is usually just important for ecology. They study different ecological studies with it. Unlike the bacteria, and the bacteria have a medical importance because they cause diseases. So the other volume 2 domain is the bacteria itself. Oh, can you tell me what is this? Which one? Clusters, C. Yes. What if it's a chain of spherical structures? Streptococcus. What do you think about this? It's false because archaea does not have a cell wall. Okay, so that's it for this chapter.